can I take you to have a bus with your neighbor to talk about, don't give us away, talk about what we learned in our previous lesson. How does water enter the plant? How does water get out the plant? Okay, so you can.
Wow, thanks.
Okay, so a question has been asked for J3 topics for this year's mid-year exam, right? How do you expect to see it? Okay, number one, it can be integrated into this year's topics. Number two, you may see it as it is, like okay. You may see your MCQs. All I can say is that whatever we are teaching this year, you will expect more questions from it, right? It's only fairer because we want to see if you understand whatever you learned from this year to a greater extent. Okay? Yeah. I'm not saying don't study last year topic. Okay? What I'm saying is that you should expect more from this year. Okay? You should be able to speak. You look quite direct. Yeah. All right. Okay. Okay, I would say for things like biomolecules, right, you should expect it quite integrated. Because by now we expect you to understand that biomolecules can be found everywhere. So something related to proteins, right, can be found in almost any chapter. Right? Because every protein got the same structures. Right? Every enzyme also has the same structure. So you can expect it that way. I don't know. Okay, let's do a quick recap. How does water enter the plant? Sorry? You give me what? Okay? Wrong. I was looking at it. Sorry? How does water enter the plant? Then wrong next. Okay, sorry, just one. Sure, okay, a transmembrane. Will you elaborate what this means, transmembrane route? Yeah. That's all. So how does it travel? Well, it travels through from cell membrane to cell membrane. So if the water has passed through the cell membrane, that's transmembrane route. Okay? Function? Symplastic. Okay, will you elaborate?
Water travel from the ureter via the plasma to the cell membrane.
Right, okay, so you mentioned the word plasmodesmata. So where's the plasmodesmata? You find that actually connecting cells, there are gaps. Right? They connect cell membrane to cell membrane. So water can also move through that pathway. That would be symplastic. Okay? Okay, last one. Sanjeev. Thank you. Can you elaborate on this? Ecoplastic. Through the cell walls. Okay, very elegantly phrased. Just through the cell walls. So if I would now draw a layer of cell wall, okay, wrapping around all these plant cells, the water will be flowing through the cell walls. Thank you. But eventually, they will also enter the plant cells because they will hit a waterproof layer called a Capillary Action. So eventually, no choice is still to enter the cell. Okay, so there are three ways water can enter the plant. Let's talk about how water moves up the plant. Also three ways. How does water move up the plant? Just one. Sure, capillary action. What about capillary action? In your own words. Right, okay, so you use the word adhesion, cohesion, surface tension. If we were to look inside a capillary, a capillary is nothing more than a tube, a very thin tube. You'll find that water molecules can adhere to the sides. Okay, they can stick to the sides of the tube. They can also adhere, there's also cohesion between the water molecules, each other. Right? So if I were to draw water molecules, let's zoom in. If they attracted to each other, that's cohesion. If it's attracted to the walls of the tube, there's adhesion. Okay, so cohesion, adhesion. These two things can help it climb up the tube. We call that capillary action. The action is the action of climbing up the tube. Alright, so what's surface tension? Actually, one would say surface tension is at the very surface of the water, there is what's holding them together at the surface is hydrogen bond. We can refer it to surface tension. You find sometimes very light things kind of stay on the surface of water, don't break through because of that surface tension. Right, yeah, the activity. Right. Oh, you're still remember that. Yeah. Okay. Number two. What other mechanisms can help water move up the plant? Read root pressure. Okay, in your own words, how will you describe root pressure? The water, what do you say? Right, okay. So if there are, here's the root cell, you're saying that the water potential is different. Then water will enter the root. Yeah? Okay, so what will be the water potential outside the root? Okay, so we're working with high water potential outside, low water potential inside. Okay, that's one way to look at it. Then, how will the water eventually get into the xylem? So here's the xylem. Here we want water to move into the xylem and then up the plant. You or anyone around who you want to add up? Okay, so the roots also draw in solutes. Sure, via diffusion, also can be via active transport. Okay, and then when there's high solute inside, then water potential will be very low, as Megan pointed out. Okay, so the root hair cells right, need to have a lot, to be very salty, then water will go in. Okay, so that's the other layer you want to add. Right, okay, then this solute will be passed on to the cells, eventually pumped into the xylem also. The idea is that we want to make the xylem high solute. If there's a lot of solute inside, the water potential will be low. This will help to draw more water from cell to cell into the xylem. Okay? We are really just making a very salty xylem. If I want to relate this to a simple experiment, I have a beaker filled with plain water, and I have a test tube that has salt water inside. Sorry, a very porous test tube that has salt water inside. You find that water wants to move in, because here is high water potential, inside is low water potential. The water level moves up. This is like a pressure pushing the water in. And that's what's happening in our the xylem and the root hair cells. The last mechanism that helps to bring water up the plant, this one is the most powerful force. Okay? Okay, thank you, Ned. Transpiration pool. What are the factors that can affect transpiration pool? Okay, anything that affects rate of evaporation can affect transpiration pool. So name some factors. Wind, humidity, temperature, surface area. Right? Okay? Plants have if they want to reduce the amount of transpiration taking place, they could close their holes, the stomata. That's one way it could regulate the amount of water vapor being lost. Okay, so broadly, these are the mechanisms. Today, we're just going to complete this chapter by looking at how plants transport sugars. And it's not quite what you expect. Yes, Debbie?
Fruits are salty, then why are carrots not?
Oh, okay. If fruits are salty, why are carrots not salty?
The solutes are not salt, like they are salts, but they are not the salts.
Okay.
Isn't the fruit like the story of the start? Oh, well, the carrots are
Okay, so what? Okay, so what is a carrot? Okay, is a carrot a root? Okay, it's a root vegetable, right? So why isn't it salty? Okay, I would say that when I say the word salty, I really would just try to oversimplify it so that you can understand that there's a difference in water potential. But I don't think it's salty to that point. And also, as Rahul said, the salt that you are thinking of, we are thinking of, when I say salty, we are thinking of like table salts, right? But there are a lot of other things that can affect water potential. For example, you can draw out minerals like sodium ion, potassium ion, not quite the things that you expect to make things salty. Yeah. But that's a good question. You bit into raw carrot and all. It's sweet, right? Okay, and that relates to what we're going to talk about today also. How plants transport sugars to the rest of the body. Okay, not body, plant. Wait, have we talked about what happens if we chop out the leaves? Oh, no, that was Ray's question, right? That's a very good question. Have we talked about what if we chop out the roots? Okay, because we are talking about a system. This is after all a transport system in plants, right? When we look at transport system in humans, remove a certain part and you can almost predict the outcome. For example, if we remove the heart of the transport system in humans, what will happen? die. But then something quite interesting. If we remove the organ of the leaves, right? Remove the leaves from the plant, as discussed yesterday, technically water can still move. What if we chop off the roots? Okay, so here's the plant. Then we chop off the roots. Then we just insert this part into water. Could water still move up the plant? Possible? Via what mechanism? All three? Oh, right, okay, so you're saying, when we talk about root pressure, are you saying that technically there are still other cells that can put solutes in? Yeah? Okay, that is still possible, but the reason why it's called root pressure is because a lot of the solutes are pumped in from the root area. Right? And that's also where all the solutes, the mineral salts are absorbed from the soil. Right? So actually that's where the pressure really primarily comes from. Could solutes still be pumped in? Still possible, the cells that are surrounding the xylem, but not as much. So I would say this one, the root pressure will be much lower, but can still survive. I mean, after all, we've had this experience before. You bought flowers before, right? No roots, right? Put in water, still can survive, right? It's not like it dies immediately. So you see, our plants are really resilient, right? For us, we chop off one arm, right? Bleed to death, right? But for plants, right? Yeah, not blood. And then the movement is also seen not mind-bendingly fast. This is the class we talk about again. How to tell whether you chop a vein or a vein. Oh, this is not a class. Okay, so you know, right? Transport in humans. How do you know if you accidentally cut an artery or vein? Okay, so, which artery? Which blood vessel you cut, right? Blood will squirt out. Okay, let's do the blood pressure. So which blood vessel, if you accidentally cut, will have very high blood pressure? The artery. Remember? Artery carrying blood away from the heart. So actually that one, the pressure is the highest. So you know you're in deep danger if after you see you fell down, right, the blood not just oozing out, it's squirting out. Okay? If it's squirting out and it follows your heartbeat, right? Okay, when I say squirt, it doesn't mean it's like a fire hydrant or just like non-stop. It will look something like squirt, because it's following your heartbeat. Yeah? So that's one thing you can take note of. Another thing is you look at the color of the blood. If the color is a bright red, that means it's high in oxygen. That means it just came from the heart. It just came from the lungs. If it's deep red, like a dark maroon, that's likely your veins, and it's likely going to be oozing out. Okay, the good part is that the design of our body, a lot of the blood vessels that you can see, right, are veins. The ones on the surface are veins. Arteries tend to be much deeper, embedded in below the muscles, right? More protection. So usually the things we cut are veins by accident. Okay? Right. So I would say that's why sometimes plants will store things in different parts of the plant. After some winter time, right, a lot of plants will shed their leaves. It will that's why it cannot last too long. Right, must store enough. Today we're going to talk about how plants transport sugars. Good thing about this is that there's only one process, okay, on how it transports sugars. And you'll be surprised, actually, it really relies on the xylem vessels. We used to think that the phloem just transports sugars by itself. But in actual fact, it relies on the xylem next door. Okay? I'd like you now to spend some time reading page 10 to 11 on the textbook first. You should also see a diagram in your textbook. Okay, we are at 407 now. After this, I'll return you your quiz. Warming blood. Not too bad, I'm fine. I think you all tried your best. Okay, so I'd like you to read page 10 to 11. Try to because I'm going to show you a video next to an animation and show you what's happening. Okay, so some silent reading time. Alright, okay, okay, fine. So 7 minutes. Yeah, it's silent, huh? I'm just going to play this remote. Can you highlight anything that you are clear about? around 2 more minutes. Okay, all done. Okay, let's complete this chapter. Yeah, you today feel like you're in a better mood. Okay, let's look at whiteboard. I set up an experiment. Here are 2 beakers, each one filled with water. No, this is not electrochem. Like, I don't know my electrochem, so this is a tube. Okay, and these are semi-permeable sacs. Okay, semi-permeable sacs. Inside here, high amount of sucrose. Here, low amount of sucrose. Okay, a quick buzz. So this is container A, container B. Do you think water will move in this direction or move in this direction? Sorry. Maybe I rephrase the question. Do you think water will move from A to B or B to A? This is A, this is B. Okay? Okay, a quick discussion. Do you think water will move from A to B or B to A? And then why lah? Okay, must be explain why. Okay, we just purely use like water potentials, you know. Okay, will water move from A to B or B to A? Okay, so will you go this direction or will you go this direction? What do you think? Okay, I put this for a reason, right? The sucrose. Okay, then maybe add one more layer. This sucrose cannot come out. It will stay in there, okay? Only water can move in and out of these pores. Okay? Only water can move in and out of these pores. Okay? It's semi-permeable in that sense. Only water can move in and out. The sucrose cannot leave. Okay? So, do you think you move from A to B or B to A? Okay? Those who say A to B, you raise your hands. Okay, those who say B to A. Okay? So quite balanced, huh? Right. Why do you think B to A? Who says B to A? Raise your hands. Okay. So, here, why do you think B to A? Okay, water potential at A. When you say A, you mean inside here? Okay, inside here is lower. So inside here is low water potential. True? True, huh? Okay. Then here is high. Right? Okay. So water will move from here to here. Oh, wait. Yeah, that makes sense. Okay. Who says that water will move from A to B? Okay. You think? Why do you say so? Okay, so, you think is saying that if you compare this and this, right, it's high water potential also, right? You say this gradient is way steeper, so you expect water to move into here. Okay, how about this one? Okay, it's less steep. Okay, fair enough. Yeah. Right. Okay, so actually can water be drawn in here? Okay, it's not possible, right, for some water to be drawn in here. Yeah? Possible? Yeah. So in reality, what happens is this one. Okay? Water does actually move in this direction. How does it happen? Actually, because of this steep gradient, a lot of water will move in first. And as it moves in, right, it kind of pushes the water in this direction. Yeah? But in doing so, as the water is being pushed in, because there's a high pressure here pushing it this way, it actually can also move the sugars along. Okay, I bring up this experiment because this is kind of how our phloem works. Sugars are being moved in the phloem with the help of movement of water. And we will watch how this happens in this video coming up. This video is trying to make sense of what you see on this page of your textbook. If you scroll the way down, if you read this page, phloem loading and unloading, and then you look at this page over here, this will be a great place to take down notes as we watch the video coming up. It's trying to make sense of whatever you see here in this picture. It is an animated version.
The vascular system of plants has two transport tissues called xylem and phloem. Xylem transports water and minerals, while phloem transports a variety of dissolved substances, including sugars and amino acids, throughout the plant. Water in the xylem always moves up in the direction from the roots to the leaves. At the leaves, water evaporates. This evaporation, called transpiration, creates tension at the top of the water column in the xylem and causes water to be drawn upward from the roots. Unlike in the xylem, the solution in the phloem can flow upward down the plant. The direction depends on the concentration of solutes in the phloem. A model called the pressure flow model describes how sucrose concentrations determine the direction of the flow. Two types of cells, source cells and sink cells, play roles in the transport of solutions in the phloem. Source cells produce sucrose and load it into the phloem. Source cells are typically photosynthetic leaf cells, but they can also be other cell types. For example, root cells with large stores of carbohydrates can act as source cells by releasing these carbohydrates into the phloem. Sink cells are any cells that unload sucrose from the phloem. Sink cells are found throughout the plant, but are abundant in roots for developing fruits and shoot tips. Sink cells typically cannot meet their own needs for carbohydrates and must import them from the phloem. The sucrose into the phloem. As the concentration of sucrose in the phloem increases, water enters the sieve tubes by osmosis. The water enters from any surrounding tissues, such as the xylem, that have lower solute concentrations than the phloem. As water flows into the sieve tubes, the turgor pressure within the sieve tubes increases. This pressure forces the sugar solution through the sieve tubes to regions of lower pressure. The solution passes by sink cells, and these cells take up sucrose. As the sucrose concentration decreases, water flows by osmosis out of the phloem to regions of higher solute concentrations. The loss of water, in turn, decreases the pressure in the phloem and pulls fluid from other, higher pressure regions of the phloem.
So we just pause here. The idea is the same. At regions where there's a lot of sugar and it's being pumped into the phloem, you'll find that you will end up drawing a lot of water from surrounding areas. For example, the xylem. And as water enters, it kind of creates a high pressure in that region that will force the sugar and the water to go somewhere else, where there's less sugar. Right? So actually, for the phloem to work, it would seem that it needs water. It's not just sugars flowing through the phloem. There's water involved. Okay. Next, we will see the same experiment.
This apparatus of beakers and funnels simulates the flow of a sucrose solution in the phloem of a plant. The funnels and connecting tube represent a sieve tube of the phloem. Differentially permeable membranes cap the funnels at the source and sink ends, allowing water, but not sucrose, to cross. Let's consider an experiment in which each funnel contains the same low concentration of sucrose. Water diffuses by osmosis into both funnels equally. Since neither funnel takes on more water, there is no net movement of the solution in the tube. As internal pressure builds, water no longer enters either funnel. Similarly, in a phloem sieve tube that contains equally low concentrations of sucrose along its length, a small amount of water enters the phloem by osmosis. However, the water enters equally along the length of the sieve tube, so there is no net movement of sucrose. In an experiment in which one funnel contains a high concentration of sucrose and the other contains a low concentration, water initially diffuses into both funnels, but soon only diffuses into the source end. As water flows into the source end, the solution is pushed through the tube and water is forced out the other end. Now consider a sieve tube that contains a high concentration of sucrose at one end and a low concentration at the other. Water initially flows by osmosis into both ends, but flows much more abundantly into the source end. This incoming water at the source end pushes the sucrose solution through the tube.
When I was a kid, I used to think the sucrose are moving via diffusion. Actually, diffusion as a process is not fast enough to bring the sugars. So we need something that creates a high pressure for things to flow a bit faster. And that pressure can come from osmosis, lots of water coming in to push it along. And then that's the process called, if you look at textbook, it's called, you can call it pressure flow or bulk flow. Okay, let's take a pause here for you to take down some notes. Yeah, anything new that you picked up? Yes. Oh, I wouldn't say so. I would say that actually the phloem already has water, but depending on how much sugar is being put inside the phloem by the tissue surrounding it, then it can end up drawing more water in. Upwards, for example. Right. Okay, but then you have to think of it as, okay, so let's say we have a phloem over here. Then you are saying it's already filled with water, right? Yeah? like only when we put sugar in, then water will be drawn in, right? Okay, so in reality, the flow already has water. In reality, there are likely some solids inside already. Okay, it depends. So for example, let's say we have a leaf over here that makes a lot of sugars, right? That will be stored inside the leaf cells. If you want to transport it from one place to another, we will first pump these sugars in here to create a lot of sugars in this region. Now because the water potential here is very low, also are you asking when the water go up? Oh, right. Oh, it's interesting. Let me think. Why doesn't the water just go up? Okay, right. Okay, let's think what requires. We think that water will move up, right? But remember, if we say water move up, right, we're thinking along the lines of osmosis, right? Right? Osmosis requires a barrier of some sort, like a semi-permeable membrane. Water would move up if, let's say, right, there is a barrier over here that only allows water to move through. Right, but the sieve tube, right, actually the holes are big enough for both sugars and water to move. So what would happen, actually, okay, it's not osmosis, but diffusion. This sucrose over here will slowly spread out and diffuse. Yeah? But this diffusion is pretty slow. We need something that will help it move a bit faster. And for it to move faster, water can move in from other cells next door, perhaps the xylem. Okay, but here's the interesting thing. There is cell membrane separating the phloem and the xylem. This cell membrane is that semi-permeable wall. That's why water will move in. Okay? So water move from xylem to phloem because separating them, not only is there cell wall, there's a cell membrane. There is semi-permeable. Yeah? So if we require water to move in a certain direction, we actually do require some sort of semi-permeable membrane for osmosis to occur. Actually, without membrane, no osmosis, you expect diffusion instead. Okay, now let's spend some time exercising our brains. Here is an interesting experiment I'd like you to take note of. If you want to, you can even screenshot this experiment and put it into your notes. Okay, it's still on the same tab, tab 2 of SLSC. Okay, this experiment is really interesting experiment. Let's see if we can make sense of it. There are 3 different parts. Okay, I'm just going to draw a simplified model. And then there's a flower here, right? Okay, so I'm going to replicate this. In the meantime, you can take down some notes, or you can also try to make sense of this experiment first. Yeah, I'm lazy. Where am I missing? I'm missing like one more thing, right? Okay, let's try and make sense of this experiment. In question 1, radioactive isotopes can be used to track movement of sucrose. What is sucrose made up of? It's made up of carbon. Where does carbon come from? CO2 in the air. Right? Okay, so first we must understand that CO2 in the air is used to make sugars. If we can make the CO2 radioactive, or rather we choose isotopes that are radioactive, then we can track its movement inside the plant. So this experiment is an interesting one. In A1 in your diagram, they wrapped A1, the leaf, and then they put radioactive CO2 over here. The second experiment, they wrap the leaf in the middle, and they allow the leaf, this particular leaf to absorb radioactive CO2. And then this one over here, they wrap the lower leaf, allow it to absorb radioactive CO2. What does this mean? It means that any sugars being made by this particular leaf will be radioactive too. Okay, I'm going to use red to represent any sugars that are radioactive. So, all the sugars made here are radioactive. All the sugars made here are radioactive, radioactive. Other parts of the leaves also receive CO2, but any sugars made are not radioactive at all. Okay? Yeah, so that's the idea of this experiment over here. But the beauty of this experiment is that now I can, in a sense, track where this radioactive sugar might go. And that's what this experiment is trying to show you. If we look at experiment number 1, we find that this radioactive sugar, on one hand, a lot of it remained in the leaf, and on the other, a lot of it went to the flower nearby. Okay, a lot of it went to the flower nearby. And some of the stem parts over here. This experiment, we find that the sugars can be found being transported along the stem and up to the flower region also, and also down to the roots. Right? Imagine if this is a carrot, all the sugars also may be there. Last experiment, we find that the sugars from here are transported along this stem and also to the rest of the roots. So let's try out this question. Why do we see no radioactivity detected in other leaves in A2 and B2? So A2 and B2. Why don't we see the other leaves being radioactive with radioactive sugar?
Because the leaves are not sinks.
Yeah, it all is
Possible? Yeah? Okay, because the other leaves over here, they make their own sugars using other CO2. Right? All the leaves are sources. Source, this is a source, this is a source. So, and this is also a source. This is also a source. We don't find sugar being transported from source to source. Yeah? It's like, why would one factory producing cookies transfer the cookies they make to another factory that also makes cookies? They want to transport it to the shops where they don't have cookies. Yeah? So that's why we find other leaves not having this radioactive sugar, because it's just not transported there. They can make their own sugar. Or in other words, they are not sinks, they are sources. Okay, I'm not too concerned about where you write this. You can either type it here, but if there's more value, honestly, maybe you want to copy paste this experiment to your notes of your OneNote, and you can write down your responses there instead. That may be more useful to you, up to you. Okay? But it's important to understand this experiment. Like the Messerson and Star experiment is one important experiment, this is an important one also. Okay? It can take some time now for you to make sense of the results. Okay, feel free to talk to your friends. Any questions you have, feel free to bring up to me also. Some of you are typing into, a lot of you are typing into SAS, it's great. You may also want to copy your response and put it in your OneNote somewhere. it really depends how you revise. If you do look at SLS again, then you revise, then okay. Okay, while you're at it, based on this experiment and where the radioactive sugars have been transported to, can you tell which part of the plants are sinks? Can you tell? Which ones are sinks? Right. The tips are. Okay, the bottom tip or the top tip. In other words, the roots are sinks. Roots are sinks. These are sinks. These are sinks. Why are they sinks? They don't have chlorophyll, cannot make their own, cannot absorb light, cannot make their own sugars. No? Flowers can be sinks also. Why would flowers need sugar? Right? What purpose may flowers have for sugars? Could be for pollination. We require sugars to make nectar to attract all the pollinators. Yeah? So flowers can be sources. This flower may eventually wilt, and then out comes a fruit. You may need you may need sugars there also, so that can be a sink in itself. One thing's for sure though, the radioactive sugar is definitely not transported to any other leaves, because leaves are sources themselves. Okay, so that's the beauty of this experiment. It actually can tell you where the sources are and where the sinks are of a plant. The last question over here, explain why radioactivity was detected above leaf level in B2 but not above leaf level in C2. Okay. How come in this experiment we don't see sugars being transported to the flower on top over here? But over here, this leaf over here is transporting sugars here as well as here. Why not? Could it be gravity? Could it be that it's too far away? Yeah? I would say a combination of many factors. Could be that it's too far away. Could be that you have to work against gravity. Maybe that force of gravity may be stronger than the pressure that's bringing the sugars there. Or another way to look at it, this flower here can get sugars from the leaves nearby. So no need to grab sugars from a leaf that's very far away. Distance also plays a role. It's just more efficient to grab from a source nearby to immediately transport sugars to the sink nearby, rather than I mean it's economics, it's logistics. Yeah. Now, why do I import things from far away when if my own country can produce its own things? Yeah. Energy, our best. Last question before we close out the chapter. Explain why it is important for C-tube elements to retain the plasma membrane, unlike the xylem. Okay, why do fluor need cell membrane? It needs it so that osmosis can occur. Okay? Fluor need to retain their their cell membrane so that osmosis can still occur. So that water can move in and create all that thermal pressure, and water will move along. Okay, so it needs a cell membrane so that we can create a semi-permeable membrane for osmosis to occur. Okay, and then we take a short pause for you to digest all of this, because we come to the end of transport and plants.
Oh, you stopped already? Okay.